Chapter 4, Biocatalysis. So there are 5 subtopics for the chapter. The first one is the properties of enzymes. Second one is mechanisms of enzyme action. And then followed by cofactors, inhibitors, and finally classification of the enzyme. So first is 4.1. There are two main learning objectives for properties of enzyme. The first one is you have to be able to define enzyme and then describe the properties of the enzymes. So there are 6 properties of enzymes in total that you have be able to explain in the exam. So what is enzyme? So the definition of enzyme is a globular protein that function as an organic catalyst that speed up the rate of metabolic reaction by lowering the activation energy. So kalau soalan minta awak define enzyme, awak kena ada semua point-point ini baru boleh dapat markah untuk definition. So in general, definition ni kita boleh bahagi kepada 2 markah ataupun 1 markah. Either way, definition tu kena ada semua point ini baru kamu dapat 1 ataupun 2 markah. Tak boleh separatekan point yang ada dekat dalam definition ini. Maksudnya kalau soalan tu suruh awak define dan dia bagi 1 markah saja, so awak kena jawab the whole sentence, iaitu starting from enzyme is a globular protein sampailah by lowering the activation energy. So the whole definition kamu akan dapat 1 markah. If the question give you 2 marks for the definition, awak kena pecah 2, iaitu the first mark will be given if you mention enzyme is a globular protein that function as an organic catalyst. Itu markah yang pertama. Markah yang kedua adalah bila awak sambung that speed up the rate of metabolic reaction or chemical reaction by lowering the activation energy. So ini markah yang kedua. So the point is, whether the question asks for 1 marks or 2 marks definition, the answer is the same. So, in the definition, the first main point is enzymes are all globular protein. Awak tak boleh simply mention dia adalah protein. Kena cakap dia adalah globular protein. So, what is globular? Globular maksudnya dia globe ataupun spherical shape. So dalam gambar ni saya ada tunjuk it's a globular shape. So itu maksudnya globular shape. Kenapa mesti mention globular? Sebab protein ada 2 classes. So the first one is globular protein. Second is fibrous protein. So for enzyme, it is made up of globular protein, bukannya fibrous. That's why kena cakap what class of protein that make up an enzyme. So itu yang pertama. Second, that function as an organic catalyst ataupun biological catalyst. So catalyst ni sepatutnya kamu boleh ingat, kamu dah belajar semasa sekolah menengah dahulu ataupun semasa chemistry sekarang. So catalyst is any compound, nutrient ataupun chemical that can speed up any kind of chemical reaction. So itu adalah catalyst. So catalyst adalah sesuatu yang awak dah belajar. So sepatutnya kamu dah ada idea dah apa yang masukkan dengan catalyst. Cuma bila kita bercakap pasal enzyme, catalyst yang dimasukkan adalah dalam bentuk globular protein. Sebelum ni awak belajar, catalyst itu mungkin dalam bentuk chemical ataupun nutrient seperti magnesium, seperti zinc. Itu example of chemical catalyst, bukannya biological ataupun organic catalyst. So enzyme adalah a version of a catalyst tapi dalam bentuk globular protein. Okay, so maksudnya bila awak dah faham apakah itu catalyst, kita tahu catalyst function dia satu saja, iaitu untuk speed up chemical reaction. Macam mana dia boleh speed up chemical reaction? By lowering the activation energy required for the chemical reaction to take place. Untuk biologi, chemical reaction itu kita panggil sebagai metabolic reaction. So metabolic reaction is one type of chemical reaction that happens in our body. So next, activation energy pun kamu sepatutnya boleh recall from your previous lesson in high school. Okay, iaitu activation energy is the minimum amount of energy required by a reactant. So in biology, under concept enzyme, the reactant kita panggil sebagai substrate to start a chemical reaction. So maksudnya untuk any chemical reaction berlaku, untuk satu chemical react with another chemical, dia perlukan a certain amount of energy. Dan energy itu adalah dalam kadar minimum. So the energy is we call activation energy. So without activation energy, chemical reaction tidak akan berlaku di antara dua reactant tersebut. So in this case, di antara dua substrate tersebut. So nothing will happen. So for a chemical reaction ataupun metabolic reaction to happen, we need to provide them with a certain ataupun with a minimum amount of energy that we call activation energy. So itu adalah requirement untuk all chemical reaction. Okay, so what enzyme or in general catalyst did is it can reduce, it can lower the activation energy so that lower amount of energy is required to start a chemical reaction. So apa yang enzyme buat tadi kita boleh nampak dekat dalam gambar rajah ni. So ada dua graf kat sini. Yang pertama adalah graf yang without enzyme ataupun without catalyst. So in general without catalyst pun boleh dekat sini. So without enzyme or without catalyst, kita nampak this is the amount of activation energy required untuk start chemical reaction yang color merah ini. So energy daripada reactant, energy yang ada dalam produk. So dia perlu kita tambah lagi energy by this amount yang kita panggil sebagai activation energy, baru reactant tersebut akan bertukar menjadi products. Okay? Tetapi bila ada enzyme or catalyst, the amount of energy required is lower. So the amount of activation energy required is much lower compared to the one without enzyme ataupun without catalyst. So kalau activation energy tu lebih rendah, so maksudnya chemical reaction ini lebih mudah untuk berlaku. Sebab kita kena letak lower energy pun reactant itu boleh bertukar menjadi products. So this is how an enzyme or a catalyst can speed up chemical reaction. Dia bukannya menambah jumlah chemical reaction. No. The amount of chemical reaction yang akan berlaku with or without enzymes are still the same. Jumlah reaction itu masih sama. Cuma masa yang diperlukan untuk perform the chemical reaction is now faster. Okay, sebab tu kita kata speed up. Dia mempercepatkan chemical reaction tersebut. So itu adalah konsep catalyst. Cuma bila kita connect dengan konsep enzyme, catalyst tersebut adalah dalam bentuk globular protein. So the concept of catalyst itself is still the same. Tak kisahlah kamu belajar masa form 5 dulu ke, dalam chemistry ke, tak kisah. Konsep dia masih sama kalau kita bercakap pasal catalyst. Cuma when we talk about enzyme, enzyme is the globular protein version of the catalyst. Bukannya chemical, bukannya nutrient yang belajar sebelum ni seperti magnesium dan juga zinc. So that's it for the definition. Next, for the properties of enzyme. So the first main properties of enzyme ada kaitan dengan definition tadi, iaitu all enzymes are globular protein which act as biological catalyst. So itu adalah property enzyme yang pertama, iaitu mesti globular protein that act as a biological catalyst. So if you remember the definition, you can get the first property of enzyme correctly. So ini sekali tadi, enzyme adalah globular class of protein. So they are not fibrous. So gambar sebelah kiri ini merujuk kepada fibrous protein. So enzyme adalah globular, bukannya fibrous protein. Second, enzyme activity is specific. So, kena cakap enzyme activity is specific. Maksud enzyme activity specific is one enzyme can only catalyze one reaction. For example, amylase, an enzyme commonly found in your saliva or digestive tract dalam usus awak. So enzyme ini can only catalyze a specific reaction, which is breaking starch into maltose. Asalkan amylase, kerja dia hanya untuk breakdown starch into maltose sahaja. So itu dimasukkan dengan property yang kedua ini, iaitu for an enzyme activity, only one enzyme catalyze one reaction. So in this example, the reaction to break down starch into maltose hanya boleh dibuat oleh amylase sahaja, tak boleh guna enzyme lain. So in another example, so under enzyme protease, so protease adalah enzyme yang function dia untuk break down protein kepada dia punya monomer, iaitu amino acids. So protease activity pun specific, meaning that it can only break down protein into amino acids. So protease tak boleh pergi ambil starch dan breakdown pergi maltose. Sama juga amylase tak boleh pergi breakdown protein kepada amino acids. Sebab apa? Sebab all enzyme activity is specific, iaitu one enzyme can only catalyze one reaction. So why enzyme activity is specific? So the reason for that is all enzyme must have active site. Semua enzyme mesti ada active site. So what is active site? So active sites is a small groove ataupun small cleft on enzyme where substrate binds to. So kalau tengok dekat example kat sini, so this is the enzyme. So dekat enzyme tu akan ada dia punya small cleft. So ini adalah small cleft tersebut where the substrate ataupun the reactant. So tadi saya kata dalam konsep enzyme, reactant tu kita panggil sebagai substrate. So where the substrate will binds to the enzyme. So dia mesti groove ataupun cleft. Maksudnya dia punya structure tu mesti masuk ke dalam. So itu maksudnya groove ataupun cleft. So all enzyme must have active site. Next, so apa kaitan active site ini dengan the enzyme activity being specific? So kita kena tengok structure of the substrate pula. So in this example, we have three different substrate A, B and C. And we can also see that each of these substrate have their own specific structure, complementary structure. So daripada tiga substrate dekat sini, hanya salah satu sahaja that have a specific structure that can fit the shape of the active site. Iaitu in this example, yang paling spesifik kepada bentuk active site kat sini adalah substrate A. So substrate C and B have a different structure compared to the active site of the enzyme. So A sahaja that can fit perfectly into the active site. So ini konsepnya kenapa enzyme activity is specific, iaitu depending on the structure of the active site that must be complementary with the structure of the substrate. So substrate lain yang tidak complementary, iaitu cannot fit the active site, will not involve in the chemical reaction. So after substrate A has bind to the active site of the enzyme, it will form a complex that we call enzyme substrate complex. So basically nak tunjuk bahawa substrate A yang spesifik kepada active site of this enzyme, and it can form a complex that we call enzyme substrate complex. So once the enzyme substrate complex has formed, it can undergo chemical reaction, and then product will be formed, and then products are released from the active site of the enzyme. So itu adalah property yang kedua. The next property is enzyme may catalyze a reversible or irreversible reaction. So in this diagram, it will show the reversible reaction. So when substrate A binds to the active site of the enzyme, it will form enzyme substrate complex. And then the enzyme will break down the substrate into products, followed by the products being released from the active site. So ini adalah example of reaction hydrolysis reaction, iaitu reaction to break down a substrate yang kita panggil sebagai catabolic reaction. So the catabolic reaction is a kind of reaction that break down substrate into products. So itu adalah catabolic reaction. Next, alternatively, a reversible reaction can also perform anabolic reaction. So anabolic reaction adalah opposite kepada breakdown, iaitu untuk buat condensation, iaitu untuk combine substrate into products. So basically it's a synthesis reaction. So untuk synthesiskan something, untuk build up something. So itu adalah anabolic reaction. So for a reversible reaction, it can either break down, iaitu catalyze a substrate, ataupun it can also synthesize a product using the substrate. So itulah dimaksudkan dengan reversible reaction. So enzyme can catalyze reversible reaction, iaitu dia boleh break down kan benda itu dan juga dia boleh resynthesize benda itu. So this is for the reversible reaction. So next, enzyme can also catalyze the irreversible type of reaction. So not all reaction is reversible. So there are certain reaction that is irreversible. So for irreversible reaction, the enzyme can only catalyze either catabolic or anabolic. Salah satu reaction saja, sama ada it break down the substrate ataupun dia akan synthesize the product. So salah satu, kalau dia boleh buat salah satu saja, so itu adalah irreversible reaction. Kalau reversible, dia boleh buat dua. So mana satu yang mana, it depends on the enzyme and depends on the type of chemical reaction involved. Yang penting, enzyme in general can catalyze reversible or irreversible reaction. The next properties of enzyme is not altered, not changed at the end of the reaction, meaning that it is reusable. So what is meant by the enzyme is not altered ataupun not changed by the end of the reaction. So kita kena nampak, so ni adalah enzyme yang asal before it bind with the substrate. So after it bind with the substrate, it will form the enzyme substrate complex where chemical reaction will occur, and then after that the substrate will be broken down into products. So the products are then released from the active site. And after the release of the product from the active sites, kita nampak the shape ataupun the structure of the enzyme by the end of the reaction is the same with the structure before the reaction takes place tadi. Okay, so itu dimaksudkan dengan enzyme is not altered ataupun not changed ataupun not destroyed at the end of the reaction. So yang berubah cumalah substrate yang akan bertukar menjadi products. Itu saja yang berubah. So the enzyme itself will not change. Dia tak akan berubah, dia tak akan bertukar, dia tak akan kena musnah by the end of the reaction. So because of this particular enzyme can be reused to catalyze another chemical reaction. So itu poin yang kedua ni, kenapa kita kata dia reusable. So since dia tidak berubah, dia tak kena musnah, it can be reused and reused and reused multiple times. Next, because of this property, dia ada kaitan dengan the next property, iaitu enzyme is only required in small amount ataupun small quantity to catalyze the reaction. So oleh kerana enzyme tadi is not altered, so it can be reused multiple times. That's why kita hanya perlukan enzyme in small quantity. So kita tak perlu letak enzyme dengan banyak untuk satu-satu chemical reaction tersebut. Kita hanya perlu letak sikit saja. Sebab sikit saja itu pun sudah cukup. Kenapa sudah cukup? Sebab enzyme itu boleh diguna pakai banyak-banyak kali. So property yang ini ada kaitan dengan the next property. So kat sini kita boleh lihat kaitan enzyme not altered dengan enzyme required in small amount. So first kita nampak kat sini an enzyme sedang bind with a substrate B to catalyze the reaction followed by formation of products. So the same enzyme is then used to bind with another substrate B and then conduct a chemical reaction and then form the product. Enzyme tu boleh lagi bind with another substrate B to form the products. So sini kita nampak the same enzyme can be reused multiple times because it is not altered ataupun not destroyed. So this is why it is required in small amount. Next, enzyme activity of enzyme is maximum at optimum pH as well as optimum temperature. So, maximum enzyme activity ni maksudnya maximum rate of reaction. So, setiap enzyme ada dia punya optimum pH, ada dia punya optimum temperature yang tersendiri. So, in this example, for this particular enzyme, the optimum temperature yang kita nampak adalah lebih kurang 37 degree Celsius. So, ini merujuk kepada enzyme yang berada kat dalam human. So in human, most enzyme in terms of optimum temperature is about 37 degree Celsius. So at optimum temperature, kita nampak reaction ini paling maksimum. So itu point yang penting untuk kita describe enzyme activity at optimum temperature, iaitu maximum rate of reaction. So what happens to the enzyme activity when the temperature ataupun pH is not optimum? So kasi kita nampak apabila dia bukan optimum, in first example, kalau temperature tu lower than the optimum temperature. So when the temperature is lower than the optimum temperature, kita nampak enzyme activity itu lebih rendah compare dengan at optimum temperature. So kenapa enzyme activity adalah rendah when it is not optimum temperature ataupun when it is lower than the optimum temperature? So semuanya because of konsep temperature, iaitu temperature will provide the heat energy that causes collision. Apabila collision berlaku, barulah boleh adanya formation of enzyme substrate complex. So that's why collision itu penting. So collision yang akan allow substrate untuk bind kepada active site of enzyme. So siapa yang boleh affect collision adalah heat energy. Kenapa heat energy boleh affect collision? Sebab heat energy yang akan provide kinetics energy for molecules to move. Maksudnya, semakin banyak heat energy, semakin banyaklah kinetics energy inside the molecule. So, apabila molekul itu mempunyai lebih banyak kinetics energy, the molecules can move faster. So, faster movement result in more collision, more pelanggaran between the enzyme dan juga substrate. So, lebih banyak pelanggaran, lebih banyaklah formation of enzyme substrate complex. So, more formation of enzyme substrate complex will result in more products form. So, itu macam mana heat ataupun temperature can affect enzyme activity. Okay, first kita tengok at low temperature, what happen? So at low temperature, kita kata enzyme is inactive. Enzyme itu tidak aktif, inactive, iaitu result in slow rate of reaction. So slow rate of reaction is because of enzyme sekarang inactive. Kenapa enzyme itu inactive? Because at low temperature, there is low kinetics energy. So low kinetic energy causes slow movement of molecules. So molekul itu sekarang kurang kinetic energy untuk dia bergerak, so slow movement, so end up causes less collision, kurang pelanggaran antara enzyme dan juga substrate. When there is less collision between the enzyme and the substrate, maka less formation of enzyme substrate complex. Ingat yang first tadi, for a chemical reaction to happen, mesti ada formation of enzyme substrate complex, iaitu when the substrate bind to the active site of the enzyme. So when there is enzyme substrate complex, barulah ada products form. In this example, when there is low temperature, apabila temperature itu rendah, ultimately there will be less enzyme substrate complex dan juga less products form. Sebab tu kita kata enzyme activity is low. So kenapa enzyme activity is low? So semua ni adalah explanation dia. Next, what happen when we increase the temperature? Tadi low, sekarang semakin meningkat towards the optimum. Tapi belum sampai optimum baru increasing from low towards the optimum temperature. So yang tengah-tengah ni lah. So increasing the temperature will increase the rate of reaction. So from the graph pun we can see that the rate of reaction slowly increase as the temperature increase. So kenapa? The main reason is the enzyme activity increases when the temperature increase. Why? Because increasing temperature will increase kinetics energy, causing faster movement of molecules, so more collision between the enzyme and the substrate, hence more formation of enzyme substrate complex. More ESC, more products form. So more products form tu, itulah yang kita katakan sebagai increasing the rate of reaction ataupun rate of enzyme activity. Next, once we achieve the optimum temperature, so at optimum temperature, enzyme activity is at maximum. Maksudnya ini adalah rate of reaction yang paling maksimum sekali. Sebab tu next one kita kata the maximum rate of reaction is achieved. Next, what happen beyond the optimum temperature? Apabila temperature to continue to increase more than the optimum temperature. So what will happen? So when the temperature continue to increase beyond the optimum temperature, selepas optimum temperature, the enzyme activity will decrease. Kalau tadi at optimum, the enzyme activity adalah maximum, iaitu maximum rate of reaction. Tapi more than that, activity tu akan decrease pula. Kita nampak dia turun semula. So kenapa dia turun semula? Kenapa the rate of reaction akan decrease beyond the optimum temperature? So the reason for this is at this temperature, iaitu temperature yang beyond optimum tadi, there are too much kinetic energy. Terlampau banyak kinetic energy yang menyebabkan bonds inside the enzyme akan starts to break. So bonds itu merangkumi ionic bonds, hydrogen bonds, disulfide bridges, van der Waals forces. So apa-apa bonds pun akan starts to break when there is too much kinetic energy. Okay, iaitu di mana temperature-nya adalah temperature yang beyond optimum temperature for that particular enzyme. So in this case, enzyme in human optimum mainly adalah 37. Kalau dia bergerak 37.1, 37.2 pun dah start kinetic energy to become too much and causing bonds to start to break. So apa kesannya when bonds inside the enzymes to start breaking? Kesannya adalah it will change the conformation ataupun the shape of the active site. So consequently, the substrate can no longer bind to the active site. So enzyme is said to be denatured. So ini term yang penting untuk describe when the conformation of the active site have changed. Okay. So kenapa dia change tadi? Because of breaking of all the bonds. Tak kisahlah bonds apa pun dekat dalam enzyme tersebut. So ingat, enzyme tadi macam mana it can bind to the substrate adalah because of their complementary structure. The structure of the enzyme dengan substrate mestilah complementary, baru mereka boleh bind dan juga form enzyme substrate complex. So kalau structure itu no longer complementary, so the substrate cannot bind to the active sites. So when there is no binding, there is no chemical reaction, there is no products formed. So situation perubahan bentuk inilah kita panggil sebagai denaturation. So once denaturation, enzyme is said to be denatured. So prosesnya denaturation, keadaan enzyme itu lepas denaturation adalah enzymes is denatured, iaitu bila conformation dia ataupun structure dia sudah berubah. So in the example, so this is the enzyme, this is the substrate. So before denaturation, the structure of the active site masih lagi complementary with the structure of the substrate. So these two molecules can bind with each other and then form enzyme substrate complex, and then reaction occur and then form products. So ini normal apa yang berlaku dalam from low temperature sampailah ke optimum temperature. Tetapi when the temperature goes beyond the optimum temperature, dia akan menyebabkan bond starts to break inside the enzyme ni, dan kesannya adalah enzyme will become denatured and then the active site has changed. Tengok bentuk active site ini no longer complementary with the structure of the substrate. Pada asalnya enzyme tu macam ni. Active site dia pun complementary with the substrate. Tapi when denaturation berlaku, iaitu breaking of bonds tadi, enzyme itu become denatured. So consequently the active site pun also change. When the active site structure sudah change, that's why the substrate cannot bind to the active site. So no binding, no chemical reaction occur. So that's why beyond the optimum temperature, rate of reaction akan starts to decrease. So semakin tinggi temperature, semakin banyaklah enzyme denature, dan kesannya rate of reaction akan finally jadi kosong. So at this point, all enzyme denatured, so no chemical reaction. Semasa tengah-tengah ni, ada setengah yang sudah denature, ada setengah yang belum denature. So that's why masih ada lagi reaction. Tetapi reaction tu makin lama makin berkurangan. Cuma at one point, apabila temperature tu terlampau tinggi dan menyebabkan semua enzim sudah denature, barulah reaction itu akan jadi kosong. So itu macam mana temperature will affect the rate of enzyme catalyze reaction. Okay, so enzyme will be denatured at extreme pH ataupun very high temperature. Ingat mesti cakap very high temperature. Jangan kata high temperature saja, kena cakap very high temperature. So enzyme hanya akan denatured at very high temperature ataupun extreme pH. Okay, so factor tadi kita tengok temperature. So very high temperature baru enzyme denatured. Kalau low temperature tadi dia tak denatured, dia cuma inactive sahaja. So jangan kata low temperature enzyme denatured. Salah Low temperature enzyme is inactive. So inactive tadi enzyme itu tidak bergerak, bukannya dia denatured, dia tak denatured. Enzyme denatured bila very high temperature sahaja. So itu tadi adalah temperature. Sekarang kita tengok the effect of pH, iaitu maximum enzyme activity is at optimum pH. So sama juga optimum bermaksud maximum rate of reaction. Okay, so for pH dia berbeza sikit dengan temperature tadi. So all enzyme function efficiently within a narrow pH range. So kasi kita nampak ada dua enzyme. For example, pepsin, satu lagi adalah trypsin. Pepsin berada dalam kita punya stomach, Trypsin pula berada dalam kita punya intestine, small intestine. So for enzyme pepsin, dia punya pH range dia adalah daripada pH 1 sampailah about pH 4. Kalau trypsin pula lebih kurang pH 5.5 sampailah ke pH 10.5, lebih kurang. So itu dimasukkan dengan narrow pH range. So all enzyme dia hanya berfungsi dekat dalam narrow pH range. So consequently, different enzyme will also have a different optimum pH depending on their particular pH range. So pepsin dia punya optimum lebih kurang pH 2, kalau amylase pula lebih kurang pH 7.2. Kalau trypsin ni kita nampak lebih kurang pH 8, dia punya optimum pH dia. So basically each enzyme ada dia punya range dia dan juga ada dia punya optimum pH dia. And at the optimum pH, dekat itulah kita jumpa maximum rate of reaction for that particular enzyme. So what will happen if we change the pH away from the optimum pH? Okay, kita gerakkan pH itu lower ataupun higher than the optimum. Lower ataupun higher than the optimum pH for that particular enzyme. So what will happen? So kesannya adalah changing the pH above ataupun below the optimal pH will change the amount of the concentration of proton H+, concentration in the solution, in the environment. Okay, ingat pH semua yang berkaitan dengan concentration of H+ dan juga OH-, tapi mainly H+ lah. Okay, so this ion ataupun proton H+ will change the ionic charge of the amino acid. Ingat, protein, enzyme adalah protein, dan kita tahu protein is made up of many amino acids, dan semua amino acids ada dia punya side chain dia. Okay, polar ke, non-polar ke, acidic ke, basic ke. So semua amino acid ada dia punya side chain dia. Okay, so consequently protein pun ada dia punya side chain since protein are made up of amino acids. Okay, so when the amount of H+ ataupun proton in the environment changes, it will change the ionic charge of the functional group of amino acid inside the enzyme. Okay, so kesannya it will cause breaking of ionic dan juga hydrogen bonds. So ingat, sama macam dekat temperature tadi, untuk setiap enzyme dia ada specific structure sebab ada bond yang akan hold the specific structure of the enzyme. So untuk pH ini, bond yang paling affected adalah ionic dan juga hydrogen bond. Sebab hydrogen dengan ionic bond ini dia ada kaitan dengan H+. Okay, so apabila jumlah H+ dalam solution itu bertukar, sebab kita move the pH lower ataupun higher than the optimum, it will disrupt the ionic charge dan causes these two bonds to break. So breaking of bonds, asalkan kita bercakap pasal breaking of bonds, kesannya it will change the specific structure ataupun conformation of the enzyme. Cuma beza dengan temperature tadi, kalau temperature tadi benda ni hanya berlaku beyond the optimum temperature. Okay? Tetapi kalau pH, proses denaturation ni akan berlaku tak kisahlah kamu move pH to lower ataupun higher than the optimum temperature. Okay, sebab tu kita kata enzyme will be natured at extreme pH. Kita tak kata very high pH ataupun very low pH sebab dia berlaku dekat dua-dua. Lower than optimum pH, higher than optimum pH, itu yang kita kata extreme. Minimum extreme dan juga maximum extreme. So kamu boleh simply cakap enzyme will be denatured at extreme pH. Tak perlu cakap very high ataupun very low pH sebab dia tak ada kaitan. So sama macam temperature tadi, denaturation will cause change in the conformation of the active site. and then consequently the substrate cannot bind to the active site. So ni yang original, so ni after process denaturation. Cuma beza dengan temperature, denaturation because of pH is extreme pH. Maksudnya low pH dan juga high pH. Dua-dua boleh cause denaturation. Kalau temperature tadi cuma very high temperature sahaja that will cause denaturation. So that's it for 4.1 properties of enzyme.